CoQ10 levels can decline with age in some tissues, particularly in skeletal muscle and brain. This reduction can impair ETC efficiency, reducing ATP production and potentially increasing ROS generation. Dietary intake and endogenous synthesis contribute to CoQ10 levels. Supplementation is sometimes explored, though its efficacy in reversing age-related decline is still an area of active research. The age-related decline in these critical mitochondrial components further exacerbates the effects of oxidative damage and mtDNA mutations. Maintaining optimal levels and function of cardiolipin, carnitine, and CoQ10 is crucial for preserving mitochondrial health throughout life. Given the significant role of mitochondrial damage in aging, researchers are exploring various strategies to protect and enhance mitochondrial function. These approaches generally fall into several categories: minimizing ROS generation, enhancing antioxidant defenses, promoting mitochondrial repair, and stimulating mitochondrial turnover, mitophagy. Minimizing ROS generation can be achieved by optimizing metabolic efficiency. Ensuring proper nutrient balance and avoiding metabolic overload can reduce electron leakage from the ETC. Research into compounds that modulate ETC activity to reduce ROS is ongoing. Enhancing antioxidant defenses is a widely studied strategy. The cell possesses an array of enzymatic and non-enzymatic antioxidants. Enzymatic antioxidants include superoxide dismutases, SODs, catalase, and glutathione peroxidases. Non-enzymatic antioxidants include glutathione, vitamins C and E, and CoQ10. Superoxide dismutases, SODs, are particularly important in mitochondria. Manganese SOD, MnSOD, or SOD2, is located in the mitochondrial matrix and converts superoxide radicals into hydrogen peroxide. Hydrogen peroxide is then detoxified by other enzymes like glutathione peroxidase. Genetically reducing SOD levels in model organisms often increases oxidative damage and accelerates aging phenotypes. Glutathione is a tripeptide antioxidant found in high concentrations in mitochondria. It plays a key role in detoxifying hydrogen peroxide and other reactive species via the glutathione peroxidase system. Maintaining high mitochondrial glutathione levels is crucial for protecting against oxidative damage. Age-related decline in mitochondrial glutathione is observed. Vitamin C, ascorbic acid, and E, tocopherol, are well-known dietary antioxidants. Vitamin C is water-soluble and can scavenge ROS in the mitochondrial intermembrane space and cytoplasm. Vitamin E is lipid-soluble and protects mitochondrial membranes from lipid peroxidation. Studies suggest these vitamins can help mitigate oxidative damage, though their impact on lifespan in humans is complex and requires further research. Uncoupling proteins, UCPs, are another fascinating defense mechanism. Located in the inner mitochondrial membrane, UCPs can dissipate the proton gradient generated by the ETC as heat, rather than using it for ATP synthesis. This process, known as mitochondrial uncoupling, reduces the membrane potential. A lower membrane potential reduces the driving force for electron leakage and thus decreases ROS production. Studies in model organisms have shown that overexpression of certain UCPs, such as UCP2 in neuronal tissue of fruit flies, can reduce oxidative damage and extend lifespan. This suggests that modulating mitochondrial coupling could be a therapeutic strategy. However, chronic uncoupling can also reduce ATP production, so a delicate balance is required. Mitochondria also possess DNA repair mechanisms, although they are less extensive than those in the nucleus. Base excision repair, BER, is the primary pathway for repairing oxidized bases in mtDNA. Stimulating or enhancing these intrinsic repair pathways could help reduce the accumulation of mtDNA mutations with age. Finally, mitophagy, the selective degradation of damaged mitochondria by autophagy, is a critical quality control mechanism. Removing dysfunctional mitochondria prevents them from accumulating and releasing pro-apoptotic factors or excessive ROS. Impaired mitophagy is observed in aging and age-related diseases. Enhancing mitophagy pathways is an active area of research for promoting mitochondrial health. The link between mitochondrial dysfunction and aging extends to the pathogenesis of numerous age-related diseases. Impaired mitochondrial function, oxidative stress, and accumulated mtDNA damage are implicated in neurodegenerative disorders, cardiovascular diseases, metabolic syndromes, and more. Neurodegenerative diseases such as Parkinson's disease (PD), Alzheimer's disease (AD), and Huntington's disease (HD) are strongly associated with mitochondrial dysfunction. Neurons are highly energy-demanding cells, making them particularly vulnerable to deficits in ATP production. In Parkinson's disease, mitochondrial dysfunction is a prominent feature. Mutations in genes like PARC2, encoding parkin, a protein involved in mitophagy, and PINK1, encoding a kinase that recruits parkin to damaged mitochondria, are linked to familial forms of PD. These mutations impair the removal of damaged mitochondria, leading to their accumulation and neuronal death. Mitochondrial toxins like NPTP also selectively target dopaminergic neurons and inhibit mitochondrial complex 1, causing Parkinsonian symptoms. Alzheimer's disease pathology involves the accumulation of amyloid beta plaques and neurofibrillary tangles. However, mitochondrial dysfunction is increasingly recognized as an early event in AD pathogenesis. Impaired mitochondrial respiration, increased ROS production, and altered mitochondrial dynamics, fission-fusion, are observed in AD brains. Amyloid beta peptides can directly interact with mitochondria, disrupting their function and transport. Huntington's disease, a genetic neurodegenerative disorder, also involves mitochondrial abnormalities. The mutant Huntington protein can interfere with mitochondrial function, transport, and calcium homeostasis. Deficits in mitochondrial complex 2 activity have been reported in HD patients. Cardiovascular diseases, particularly age-related conditions like dilated cardiomyopathy and heart failure, are also linked to mitochondrial dysfunction. The heart is a highly metabolic organ, relying heavily on mitochondrial ATP production to sustain continuous pumping. In cardiomyopathy, impaired mitochondrial respiration, increased oxidative stress, and mtDNA damage are observed in cardiac muscle cells, cardiomyocytes. These defects compromise the heart's ability to generate sufficient energy, contributing to contractile dysfunction and heart failure. Research into targeting mitochondrial ion channels, like the MitoKCA channel, to protect cardiomyocytes during stress events like ischemia reperfusion injury shows promise. Type 2 diabetes mellitus, T2DM, a metabolic disorder strongly associated with aging and obesity, also involves mitochondrial dysfunction. Insulin resistance, a hallmark of T2DM, is linked to impaired mitochondrial function in insulin-sensitive tissues like skeletal muscle, liver, and adipose tissue. Reduced mitochondrial content, impaired oxidative phosphorylation, and increased lipid accumulation due to compromised fatty acid oxidation are observed in muscle cells of insulin-resistant individuals. Certain mtDNA mutations are also associated with impaired insulin secretion by pancreatic beta cells. The progressive accumulation of mitochondrial damage with age is hypothesized to contribute to the increasing incidence of T2DM in older populations. Investigating the complex interplay between mitochondria and aging requires sophisticated techniques from various fields of biology. Advances in genomics, proteomics, and molecular biology have revolutionized our ability to study these organelles. Genomics approaches allow researchers to sequence and analyze mitochondrial DNA from different tissues and age groups. This helps identify age-related mutations, deletions, and changes in mtDNA copy number. Databases like Mitoproteome and MyTop compile information on mitochondrial genes and proteins, facilitating systems biology approaches. Proteomics enables the study of the entire set of proteins within mitochondria, the mitochondrial proteome. Techniques like mass spectrometry can quantify changes in protein abundance, post-translational modifications, and protein-protein interactions with age or disease. This helps identify which mitochondrial pathways are affected. Recombinant DNA technology and genetic engineering are crucial for creating model systems. Researchers develop cell lines or animal models with specific genetic modifications affecting mitochondrial function. For example, mice with reduced levels of mitochondrial SOD2 are used to study the impact of oxidative stress on aging and neurodegeneration. These models serve as living laboratories to test potential therapeutic interventions, such as antioxidant therapies or gene therapies targeting mitochondrial pathways. Despite significant progress, studying mitochondria presents unique challenges. Their double membrane structure and distinct genetic system require specialized techniques for isolation, manipulation, and analysis. Delivering therapeutic molecules or genes specifically to mitochondria remains a technical hurdle. However, the field is rapidly evolving. New tools for targeting mitochondrial DNA, modulating mitochondrial dynamics, fission-fusion, and enhancing mitophagy are being developed. The increasing understanding of mitochondrial signaling pathways and their integration with other cellular processes opens up new avenues for intervention. While completely halting mitochondrial decay may not be feasible in the near future, scientists are increasingly confident that the process can be slowed. By preserving mitochondrial health and function, it may be possible to delay the onset and progression of age-related decline and associated diseases, thereby extending health span. As biology students, you are at the forefront of this exciting field. Understanding the fundamental biology of mitochondria is not just about cellular energy. It's about unraveling the mechanisms of aging and age-related diseases. The knowledge you gain today is the foundation for future breakthroughs. Dive deeper into cellular respiration, genetics, and molecular biology. Explore research papers on mitochondrial dynamics, oxidative stress, and mitophagy. Consider how your understanding of these processes can contribute to developing interventions that promote healthy aging. The future of extending health span and combating age-related diseases depends on the next generation of scientists. That's you. Engage with this material, ask questions, and consider how you can contribute to this vital area of research.